Hello everyone, welcome back to our channel. Today we will discuss about the Leishmania Donovani. See here, Leishmania Donovani. It is under medical parasitology, where we are discussing about the parasite, that is parasitology. Leishmania Donovani is one of the most important parasite we will discuss. Firstly, we need to understand the location, where they are located. Leishmania Donovani basically located into reticuloendothelial system into our human body. They basically located here. It is the morphology of the Leishmania Donovani. There is a two type of form. One is emastigot form and another one is promastigot form. Emastigot form basically present into the human and promastigot form basically present into the saint fly. We have an idea regarding the host. Hosts are where basically parasites are taking their nutrition and their surveillance from the host. Hosts who are giving to the nutrition, to giving the energy, to giving the places to survive a parasite, that is called host. Basically, there are three types of host. In the another class, we will discuss the three type of host. But here you will see two type. One is definitive host and another one is intermediate host. Definitive host means where the parasite are doing their sexual replication. That is definitive host. And another one is the intermediate host where the parasite are doing their asexual replication. That is called intermediate host. Into the case of saint fly or into the case of Leishmania donovani, here the definitive host is human or and the intermediate host is saint fly. So that the life cycle are occurring within the saint fly to human beings. Basically, the parasite are belonging into two body. One is saint fly's body, another one is the man's body or human's body. Understood? That is the basic life cycle. We will discuss the life cycle in another class. It's not possible to covering everything within one class. Again, come to what disease basically form. Parasitism. Parasitism define that which kind of parasite are causing the disease. That is called parasitism or parasitism. Their relationship with our human body is parasitism. They will cause the disease. See here, Kala Azar, or another name of Kala Azar is Leishmania, Leishmania, Leishmaniasis. Understood? Like whenever the Ascaris lumbricoides are attacked, that is Ascariasis. Whenever the microfilar is attacked, that is microfilariasis. Whenever the amoeba it attacks, that is amoebiasis. Understood? That is also called leishmaniasis. It is the disease condition, leishmaniasis. What are the features of this disease? Firstly, pyrexia. Whenever the high level of fever is happening, that is called pyrexia. After that, splenomegaly, spleen become enlarged. That is splenomegaly. That is also the symptom of Kala Azar. Hepatomegaly, liver become enlarged. That is hepatomegaly. Lymphadenopathy, whenever the lymph node inflammation are happening, that is called lymphadenopathy. Understood? These kinds of symptoms are occurred or associated to the Kala Azar. Very easy, we have already covered the disease condition. Now we are coming to the lab diagnosis. That is our work. Because we are the medical laboratory professionals, we require to understand how do we diagnosis the Kala Azar. How do we diagnosis the leishmaniasis? We need to understand it. See here, I have firstly we have to do peripheral blood smear because it is holding as well as it basically belonging into the reticuloendothelial system, so that we might found the emastigot form, we might found the emastigot form into our blood, so that we have to do the peripheral blood smear. See here, A define the emastigot form. We might see the emastigot form whenever we will do the blood smear. Okay. Second, blood culture. We can do the blood culture. The media is NNN media. Whenever we will do the blood culture, we might see the promastigot form. Understood? After that biopsy, we might do the liver biopsy, spleen biopsy, because splenomegaly are occurring, liver as well as hepatomegaly are happening, as well as this disease or this phenomena or this infection basically attacks the liver, attacks the spleen, so that we can do the biopsy for taking the tissue from this particular organ. After that, see here, splenic puncture. Whenever splenomegaly are happening, we have to do the splenic puncture. We have to do, not we might do. That is the optional things we might do into our laboratory diagnosis section. We might do the puncture of spleen so that we can easily identify the hemastic God form from the spleen. After that, that's all are the non-specific. Non-specific. See here, that's all are the specific. Specific means which diagnosis or which test we are doing for a particular diagnosis or for the particular disease we are doing, that is called specific diagnosis. And another one is the non-specific. Non-specific means that is not truly connected with the disease but might be happening. We have to do the blood count, and whenever we will do the blood count, we might see that WBC level become elevated. Understood? Because infection are happening, as well as eosinophilia become happening. Okay. And one thing please keep in mind that we have to do the hemoglobin estimation. Here hemoglobin become decreases, so that anemia might be occurred. After that, we have to do the aldehyde test. That's all are the non-specific tests we might do into the leishmaniasis, as well as into the Kala Azar diagnosis, as well as into the Leishmania Donovani attack. So that please keep in mind that the scent fly and the human. Today's class up to this much. Into the next video, we will discuss about the life cycle of Leishmania Donovani. Thank you so much for watching.